Hi, everybody. Welcome to the first podcast in our regulation domain. In this first one, we are going to spend some time looking at genetics from an overall systems perspective. And this can be broken up into two sections, so you'll see a definite part 1 and 2 that we're going to work with. But the big picture ideas, how do genetics ultimately contribute to the development of an organism? And what are the ways in which we, create varied system and differentiated cells through development. So how does evolution then influence also the structure of an organism's genetic code? So these two things are kind of tied hand in hand that we're going to spend a little bit of time looking at as we go through ultimately the big picture idea here on how living things are regulated. So we're going to begin with developmental genetics. So in this first part, we're going to look at differentiation, how cells that begin with the same volume of genetic information acquire very specific different structures and functions. Why are certain genes turned on in some cells, not in others, and how is this determined? Because at one point in time we were purely a ball of cells that were identical to each other. So let's look at this from sort of this first piece. So we begin at that fertilized egg, right? That zygote, okay? And then how do we go from that single cell to a newly hatched tadpole that has very small, very differentiated cells doing something that is obviously pretty important to get us from A to B. So let's figure that out. So some of the things happen to do with cytoplasmic determinants. Okay, so substances that are present in the cytoplasm of an egg cell are unevenly distributed. This is going to begin the process of how when that first meiotic divisions happen, that some cells end up with an uneven distribution of materials that were inside that cytoplasm. And so direct gene expression will then happen in subsequent generations of those cells because you can see you have that change because you can see you have that early change. So you get from the fertilized, you can see that they're uneven in terms of the signalings that are inside that cytoplasm, and then at that first division, those two cells, you can see you're very unevenly. And this becomes really, really important in early development because this is going to set the stage and set the pathways that the cells are going to then become. So then we have induction, and from induction you have your clump of cells, right? That signals from differing surrounding cells send information to cells nearby that help to send it out down a particular genetic pathway and a differentiated pathway. So this becomes like uber important. Okay, signals, again, keep in mind these are hormones. They're acting at receptors. They influence gene expression of genes inside the nucleus. That's really important for later determination. So the determination, as I just said, the regulatory events in the genome lead towards a differentiation in the structure and the function of those cells. So once a cell, though, commits to that particular pathway, it can't uncommit. So you go from a cell that is what we call basically omnipotent, right? Where it can become anything, to a pluripotent cell, which means that it had, kind of down the pathway, but it can differentiate into numbers. But once it hits that pathway, it's gone, okay? And you can't really turn it back. We thought naturally they can't be turned back. However, there has been, and this was the Nobel Prize winner a couple of years ago, the ability to reverse that signaling and send a cell that is fully differentiated back to a stem-like cell state. Pretty cool stuff. So we'll talk a little bit more about that down the road. So here's kind of what happens. So you've got particularly embryonic precursing cells that have genes that are on, cause different things to happen. Then you got kind of how this cell is going to ultimately become that muscle cell. So it differentiates due to certain genes being turned on, a myoblast, which is the precursing gene to that fully functional muscle fiber cell. And you can see there's different effects that occur, different transcription factors that happen that act at different genes, those act at other genes, and then you get down the road differentiation that's going to occur, right? There is also pattern formation that happens. So how a body plan is established, we know as human beings, and you know this about a lot of critters, that if you're of the same species, things are generally laid out in the right place, okay? We all have our head up here. We all have our feet down there. We all have, our heart right here. So how is that happen? The actual plan established in a developing organism. And there's kind of two factors and two ways that this works. So we have positional information. So molecular signals establish, right, the body axes in a developing organism. So we've done a lot of work with fruit flies on this in understanding, different areas that determine, oh, this area here is going to become the head. This area here is going to become the abdomen. This is going to distinguish left from right, anterior, posterior, all of the different axis points, and sagittal and all of these locations that are going to send those signals. So over a long period of time, we can establish what that has been. So you get the egg, that's developing with the nucleus, right? Some cells that are there to help, provide energy, those nerve cells. So you get the fertilization, okay, and then that embryonic development, you can see it breaks down into segments, okay, and those segments then help with the metamorphosis of the bug from the larva into the pupa into the adult, okay. So pretty, time consuming to figure this out, but there are areas that you can find, and if you mess with those areas, you mess with the ultimate outcome that happens. So you can see the green, okay, there's the area that goes on to become the head. The brown becomes the thorax, okay, and the legs. You've got the orange, which is your abdomen, and these are all signaling pathways that do that. Okay, so here's an example. We've got a bicoid, okay, protein present in a differential gradient in an unfertilized egg. Okay, so this protein, okay, is an example of how this, well, I should back up. This is an example where this particular protein in different amounts at different times can cause different cells to differentiate. So cells that develop in highly concentrations of this bicoid become the anterior, okay, so anterior, just so you know, the head area, okay, anterior means front, okay, and then you have posterior behind. So anytime you're talking about something that's anterior to something else, okay, it's in front of that. So you have like your ACL, your anterior cruciate ligament, so ligaments in your knee, it's always relative, okay, I can't just say this is anterior, okay, not anterior to what, okay, so it's always about relativity. So anterior is in front, so the anterior cruciate ligament is the one behind the posterior cruciate ligament. And so those are just descriptive terms you'll see a lot in anatomy. So the head area of the organism. The bicoid controls head development, so it's referred to as a morphogen, okay? All right, so differences that are going to occur. So here you have where you've got these areas, okay? Normal and a bicoid mutant of this fruit fly larva. You can see that if there's different signalings, you're going to have the tail be produced in certain areas and not the head, okay? So this protein is really, really important for these things, okay? So here are your results. If you shift that distribution, okay, you're going to get not the right things being produced, okay? So you got the bicoid mRNA in a mature unfertilized egg. You can see where its amount, and you can see where it's in high concentration. That's the area, okay? That will then become the anterior end. So this is how we can tell from behind for the developing cells. In an unfertilized egg, okay, in a mature unfertilized egg, the bicoid in an early embryo. Okay, so you can just see the sort of, the cartoon versus the actual pictures of what that's going on. All right, so regulators, okay, so you get these developments, all these different signals that are going to be there, but there's regulation, and here's where the homeotic genes come in. Now, these were first discovered in fruit flies. Again, fruit flies have given a ton for genetics to us, and ultimately the homeotic genes won the Nobel Prize for Edward Lewis in '95. Huge amounts of animal lineages that have these, and they are highly conserved over evolutionary time, right? Highly conserved sequences, and what we call the homeobox, okay? A particular series of nitrogen bases that we use, okay? So these are really important for determining location of structures in the organism. This is a fruit fly type, okay? The mutant, it's actually a dominant problem called Antennipeda mutants, and they have legs that are in the wrong places. Basically, instead of growing normal antenna, they grow legs there instead because of signaling problems that go wrong in this mutant of this homeobox. You have an eyeless mutant, okay, because they have vestigial eyes in totally wrong places, okay? So these eyes that don't function but grow where they're not supposed to, okay? And easy to see why we can do these in fruit flies. Look at that double set of wings. Generation time is really quick. They only have 8 chromosomes. You know, there's a lot of, sort of simplistic nature that we can work with these guys and get a lot of information in a really short amount of time. So fruit flies are good for that stuff, okay? So here's the homeotic genes that we can look at in an adult fruit fly. So when you look at the fly chromosome, okay, you can see on that chromosome there are particular areas located on the chromosome that distinguish particular areas in the development of location of parts, okay? The adult fruit fly, you can see where those are located, how they look in the embryo that's only 10 hours later, okay? And they match right up. And we can see this also not only in invertebrates, but we can see it in vertebrates like in mice. Okay, it's a little bit more complicated because we have more that deal with it, more chromosomes, but you can see the chromosomes in the mouse have matching important, not only location on the chromosome, also dictates location on the organism. Okay, so where those genes are located determine signaling pathways that showcase development of the organism with the location of stuff being in the right place. And they're highly conserved. Again, these show consistencies generation after generation. They don't seem to be changed much over the evolutionary time frame. And so here we have Hox genes. So this shows 7 different homeobox expression patterns in a fruit fly embryo. This is what we call the Hox genes activity. And you can see that based on the coloring, it locates right in order, which is really kind of cool. So homology groups, these are the same. So in those fruit flies, that's what you get. So the fluorescence matches them right up in that embryo. And we'll do an activity that'll kind of showcase this a little bit more too. So differences in homeobox expressions that lead to differences in segmentation in insects. So, insects are segmented, again, the head, the thorax, the abdomen. And these different locations of these genes help to set the pathway for structures to be set in the right place. So in this first part in development, make sure you can do these, because then we're going to move right into the genomic pieces on sequencing and how we figure this stuff out. So more of a technology-based aspect. So those are the things you want to do here. If you want to pause, take care of part 1, go for it. Okay, we're going to move on to part 2. So here we go. Okay, go into the Prezi and watch that if you want to. Trust me, it's kind of funny. So back to part 2. Genomic sequencing. So how, from a technological standpoint, can we figure this stuff out, right? So process, sequencing a genome. You shred it up into bits, you sequence the shreds, and then you line it back up again with a computer. So that is it in a nutshell. Okay, so first off, we have the Human Genome Project method of what they did to look at, in the early days, Human Genome Project launched in 1990. So they, had the chromosome, the genes, linkage mapping. They had to look for when genes were expressed simultaneously, when crossing over would be more likely to separate them, and then they physically mapped them out anywhere that would overlap, and then determine the sequences from there. Okay, so that's kind of how they worked with it there. Okay, and so this from DNAi showcases how the procedure was done. So again, I'm gonna leave this here for those that want to go in and check it out. Check it out in the link to the presentation. Then you have sort of this whole shotgun sequencing. Slice the DNA up into fragments short enough for sequencing, okay? Clone the fragments, okay, either in a plasmid, do it in a vector, okay? Sequence those fragments, and then find where all the overlapping ones are to place them in the right spot. Okay, so that's kind of the whole genome sort of event to do that. Okay, so here, again, a little visual on shotgunning sequencing. Early on with the Human Genome Project, this was really expensive to do, and then over time we have been able to do far more with a lot less money. So the technology has allowed us to spend less money. In terms of the numbers of published genomes, you can see it has just skyrocketed because it's so much quicker and so much less costly to do this now. So then we have to analyze the information. So how do we make sense of all those millions and billions of base pairs? So we use computer analysis to look at this genetic information, and these databases, bioinformatics is so important to be able to look at. We've used a little bit of that early on when we went in and used some of those databases to actually sequence and align genes of different organisms, and this has made the process so much easier. So just to compare some of these genomes, you have bacteria and archaea, much smaller, the number of genes that are present versus eukaryotic organisms, how the introns are there, what exons are there, non-coding DNA sequences. So take a second and kind of peek at these with just some comparisons of how genomes vary, whether you're a bacteria or a eukaryotic, and why some of it gets a little bit more complicated as we continue to talk about this, right? So there's it, just again with some examples of some of the eukaryotes. You can see the number, the haploid genome size, okay, number of genes that are present, genes per, the number of genes that exist in terms of million base pair length. So take a second and peek. It's kind of interesting to see the variations that do exist in genome sizes with the estimated number of genes that are there. And then yeast cells. This is sort of an interactive map looking at 4,500 different proteins and how these proteins establish how they act. Now, this is something that is becoming a much, much bigger area and field of study is proteomics. It's one thing to know the blueprint for the protein, which we do with the genome. The other part is understanding the role and how proteins fold, how they interact with one another. We know proteins are so important that they're so complicated and they serve so many functions. So this alone is just 45,000 proteins in a yeast cell, okay, in a yeast organism. And you can see the interactions are so vital with the jobs that these proteins do. Okay, so that's something, that's another piece. There's also tools for microarrays where you can, I imagine down the road, sometime in probably my lifetime, definitely yours, we're going to carry in our wallet a card that has our entire genome right on it, on a little chip. You can do it now, it's just costly. So different microarrays, we can throw our whole genome right on a chip. So the Genome Project was a big deal. This sort of set the stage for us to understand how our genes functioned, how they interacted, what went on. So the Human Genome Project's been sort of established in a number of ways. So here's what it looks like. You know the drill. Here's sort of a pie chart looking at what our total DNA amounts to. Okay, and you can see that pretty much regulatory sequences, that's your coding DNA, amounts to only about that's for regulation, right? Regulatory sequences, that's only going to do 20%. Non-coding, 15. Can you find where our coding information is, like that actually codes for the proteins of our DNA? I can tell you it's the smallest pie piece up there. Just saying, smallest pie piece up there. Transposable elements, you guys have heard of these before. That's part of the repetitive DNA sequencing. Non-coding segments catalyze their own replication, movement through the genome. Barbara McClintock first found these in corn in the 1940s. This was, super amazing scientist and won the Nobel Prize in 1983. And she first saw this with the jumping genes that we saw in the corn. Why were certain corns carrying combinations of colors? And that's because of the transposable elements that happen from chromosome to chromosome, okay, and hence the jumping genes. So here's what happens. You get a piece of a transposon sequence that codes for their own replication and insertion. So this transposon creates a segment and then inserts itself somewhere else and is expressed. So that's what it is. And then you can have retrotransposons, excuse me, that kind of do everything in reverse. Transcribed into RNA, reverse transcribed back into DNA, and inserted. And they code for their own reverse transcriptase in order to do this. It's crazy stuff. So we have a small portion of retrotransposons found in primates. Those are our ALU elements that we've looked at in the PCR. Short tandem repeats, these are a little bit bigger. They can be upwards, to 500 base pairs long. This is one of the things they'll look for when, that is unique from person to person. So one of the, they'll do STRs when they're doing DNA fingerprinting. Okay, we have large scale, kind of large segment duplications. These are thousands of base pairs present in only a few copies that you have. Most are actually on the Y chromosome, so there's part of what is your Y chromosome. You have unique non-coding DNA, pseudogens, gene fragments, still trying to figure out what these do. Your regulatory sequences, those are your promoters, your enhancer regions, all of the areas that sort of flank and support the actual coding section, right? Introns, these. We have about 3 times more introns than we do exons. Okay, these are what are spliced out when we process mRNA. And then finally, there's the actual coding sequences that produce all of the different proteins our body has in 1.5% of our DNA. So think about that for a second. 98.5% of our DNA doesn't actually code for functioning protein. It is everything else that supports and surrounds that information, which is just so stinking cool. Not gonna lie. So the evolution of our genome. Let's look into this. Okay, so now that we have the data, okay, we can analyze it for patterns, right? We can look, and one of the main things that that's thought of and theorized and supported is that over time, if you share more DNA, you're more likely to be more related, right? So we have signatures in the data. So we look for things that are going to be consistent. So we have gene families. This refers to collections of genes that have very similar structures and identical. We can look how organisms share these, how they are shared within organisms. So following duplication, sequences may diverge in their structure and in their function. Okay, so we kind of get that, right? You can have identical sequence families, genes that occur multiple times in a genome. They're usually clustered together. rRNA does this, okay? We have tons of copies of rRNA because we need to make a lot of ribosomes. So that's pretty normal to have that part that's going to create the 2 components that build, that build up a ribosome. And this is pretty consistent critter to critter to critter to critter. Because we all need ribosomes, right? Every living organism does, and they're all really similar. And then you have non-identical sequences. These are related, they're not identical, but they can be found in different locations of the genome. Alpha and beta globin genes, okay, this is what create your hemoglobins. You have the alpha on 16, the beta on 11, yet they work together to create that one single protein molecule. So there's that. Okay, alterations in your chromosomes, rearrangements so that structures leave particular signatures. So if chromosomes rearrange in themselves, you can actually get consistencies that can exist here. So chromosome 2 on a human, okay, chimpanzee chromosome 12 and 13, okay. You've got locations that are very, very similar in ours of where the centromere is. You can notice that there's issues where the telomere-like sequences that exist in ours, it's almost like those 2 at one point joined. So there's a theory there that might have been what happened because of these signatures that exist. Because in ours we also have a centromere-like sequence that's just below the telomere-like sequence on our long arm of chromosome 2. Could it be? Okay, there's also things between us and mice as well. Evidence of these fusions are found in the genomes when we look to it. So these are similarities that lead us to hypotheses as to looking at evolution, looking at how life is related. Okay, and then translocations during crossing over. Okay, so crossing over points can be actually somewhat consistent. And because of that, we can look for transposable elements that can occur from crossing over between non-sister chromatids, and we can look at crossing over that occurs between non-sister chromatids of different homologous pairs, right, and have that actual transposon that exists. And these leave marks, that we can look at as consistencies. So when we compare our genomes, right, we can look at similarities and differences that provide evidence on how evolution has changed the genetic structures of different species, and give us some insight as to the maybes behind how things came to be. And this, connects back to what we did in the opening portion of the year when we looked at sequences and how we can look at the divergence based on similarities in those genomes. So when we correlate that genetic change to the fossil evidence, giving us some dating and some timelines for these phylogenetic trees, right? And that's what we can kind of look at and understand where did life diverge at different moments in time. So here's a little experiment. FOXP2 gene involved in language in humans and related to development in various structures in the brain. Okay, so let's take a peek here. Okay, so here's the experiment. 2 normal copies of the FOXP2, and then you can have, that's your wild type, heterozygote, 1 copy of the disrupted kind, homozygote, both are disruptive. So in the experiment, researchers cut thin sections of the brain, stained them with reagents to look at the anatomy under UV light. Okay. So you got your wild type, you can see what happens in the heterozygote, you can see what happens in the homozygote. So there's definite structural differences that occur, okay, when you have varying, genotypes with these genes. In the second experiment, okay, researchers separated each newborn pup from its mother and recorded the number of ultrasonic whistles produced by the pup. Okay, in the little ones, although it sounds kind of sad to think of little mice as crying for their mommy, little mice. So the wild type, who had 2 normal copies, gave out huge amounts of whistling. Okay, cried far more often than the heterozygote, than the homozygote. And in the homozygote, for the mutant, there were no whistles at all. So even in humans that we can see that it helps with brain development for speech patterns, in mice, brain development, speech patterns that exist. So very similar, right? So some way, shape, and form, we have structures that are consistent. We are connected to life. So duplications and divergence. So when genetic information is duplicated, we know that there are mistakes that can be made that can then acquire different, novel functions, right? So these processes that occur at the chromosome, they can be intragene levels of genome organization and can cause different outcomes to happen. So similarities in sequences between the human globin proteins. So you got your alpha and your beta. Okay. The idea is that there's these 2, they exist on 2 different chromosomes, okay, and there are some similarities. So over evolutionary time, the sequences can be analyzed to try to understand how far back mutations occurred, when the duplications caused there to be a divergence in the 2 that were produced, okay, that there were mutations that occurred, and we can come up with an evolutionary hypothesis based on this. Different factors of exon shuffling, right? We know that that can happen with exon shuffling. So sequence analysis can look at our current TPA gene, and I'm going to make you look up what that is. Okay, see if you can find out what that is. So results from the transposons of exons from 3 different genes. Okay, so that there was shuffling. So when we look for these markers, again, we're looking for similarities, and these similarities are giving us insight into the possibility of what of those connections. We're looking for the threads. We're looking for the bits of evidence that might show how the life all interconnects. So we have some ideas on that with what we have. So will we ever find out the answers? I don't know. We can let the evidence roll in the direction that it rolls in. The key is to look at it and take it for what it is and see if we can make sense of it. So from this, we're going to go and start looking at furthering our understanding of development, furthering how systems regulate, and not forgetting the fact that it's all ultimately, controlled by what the genes are. So in this last part for the genomics, how they're sequenced, how bioinformatics is important, how genetic events lead to the evolution of our genes and our different gene families, how duplication can lead to the evolution of new genes with novel functions, and really look towards that. So spend some time with it. I realize it's long, 2 parts. Enjoy it, and we'll talk in class for things that you feel like you need to. And if you need to go back to watch those videos too, please do. So have a good night, guys, and we'll catch you in class later.